Hello, thank you, Kennedy. Thank you all. You see, I can't see the screen here. There we go. That's better. I'm going to tell you a detective story. A detective story, an unsolved mystery, 50 years unsolved. I'd like your help in solving it. The characters up there are a wonderful Nobel Prize winner, Edward Purcell, a swimming little microbe waving its flagella, and it or its brothers and sisters and cousins have been around on Earth for about 1 billion years, billion with a B. And over on the right is one rendition of a water molecule published by George Walrafen about 60 years ago. Why am I showing you all this old-fashioned stuff when we'd all like to be terribly modern? It's because according to the best scientists today, they don't play well together. Let's try and fix that. Let me see, how do I advance the slide? Button, the middle? What are we doing here? How do I move this slide up? I think it's upside down. Oh, that does it. There we go. I am an oceanographer. I go to sea on ships. I make chemical measurements. We use lasers a lot. I once did a famous deep ocean NMR experiment, if you can imagine that. Top left corner, that is a remotely operated vehicle. It weighs about 7 tons. It's going into a test tank. We use it as a carrying platform. It takes down lasers, all kinds of instruments. We can do a vast array of chemical experiments in the deep ocean, carrying them down a device. It's got 2 robotic arms. Like robotic surgery these days, we can do clever things, which we like to do. There is, let me see, where are we? Oh, no. Yeah. How do I point the laser? I need some instructions here. Oh, yeah. There's the laser, and then we turn. Yeah.
That blinks the screen, the bottom button. So hit the bottom button again.
Okay. That's us in action. We're on a ship. There's a hole in the middle of the ship. We call it the moon pool. That is a 7-ton vehicle about to be lowered into the ocean, carrying my stuff on it. Little panel at the bottom is actually a laser Raman spectrometer. We package the laser, everything else in there. The light goes out through a bunch of optical fibers out to the front of the vehicle. We point it around, and you can point the laser anywhere you want and get a signal, just like Star Wars. Bunch of optical fibers. We communicate over 4,000 meters of cable through high-speed internet. It's all good. I keep screwing up on this. Where do we go? Nope, I got it wrong. Here we go. We're back again. I could have used some advice here. Few words about the ocean. 92% of the water in the world is in the oceans. 92% of all the greenhouse gas generated heat, the problem of global warming and climate change, the heat goes in the ocean. And about 92% of all ocean scientists have no idea that water has any molecular structure. It's a disaster. That's our job. Don't take my word for it. Here's a book. This one's Environmental Fluid Dynamics. I could have brought a library. You will look in vain for a hydrogen bond or a water molecule anywhere in here. I'll bring you a book on aquatic chemistry, a stack of books this high. Water molecules aren't there. Why did we choose Raman spectroscopy? Why did we choose lasers? Because they work well. So here's a little quote from Sir Chandrasekhar Raman. That's why the technique is called after his last name, not his first name. He took a voyage from India to England, went through the Mediterranean Sea, and loved the blue color. He became entranced. He had an epiphany and he wanted to devote his life to the molecular scattering of light. He probably made a mistake. What he was seeing, I think, was the color change caused by chlorophyll, fluorescence, makes it look reddish. If I tried to do a Raman spectroscopy experiment in water lit by sunlight, it would kill the signal. If I tried to make it a measurement in water containing chlorophyll, it would kill the signal. But never mind, we will adopt him as an ocean chemist because he's a nice guy and he wrote some nice stories about him and music. What's the problem we're going to talk about? How did we all get here? How did I get here? How did you get here? Kennedy, how did you get here? Not by walking, not by bike, not by the courtesy shuttle. We all got here, as Francis Arnold said, when a sperm cell had to swim to fertilize an egg. How does it do that? We know about DNA. We've heard a lot about it. Scientists can't agree on it. What they can't agree on is the swimming. You might think it's easy, but nobody agrees on that. So here's a picture. There's a quote from Sir Geoffrey Taylor, a world-famous fluid dynamicist, who was investigating the swimming of bovine sperm. And he noted that the viscous forces in the liquid were thousands of times greater than the inertial forces. When you swim, you're doing this, you're pushing the water backwards. When a fish swims, it's doing that. When these guys try to swim, they can't do it. To them, this looks as thick syrup. You imagine trying to swim in thick syrup, you can't do it with the apparatus you've got. There is a sperm cell, and there's an engineering analysis of how it manipulates that long flagellum. You'll notice there it seems to be flying through air. It looks like a NASA spacecraft. It's not really swimming through water, but it's supposed to be. But it rotates this, and they try and create the forces which drive it. There is an E. coli bacterium, which is what Purcell investigated. It has 4 flagella. I think the record for number of flagella is 7 now. How do they do it? They twirl those around. This has a complete circular motor, supramolecular chemistry, well before Fraser started. And those things rotate, and that's how they move. I'm going to get this right now. There we are. Here's the story. Famous guy, Edward Purcell. Love him. I found out yesterday that our friend Richard Zare knew him personally when he was at Harvard. Great guy. He loved the theater. Writes beautiful papers. He got the Nobel in 1952 for the discovery of nuclear magnetic resonance. He shared it with Felix Bloch of Stanford. But my favorite paper of all time is his 1954 paper with Carr and Purcell. Wonderful, beautiful paper. I advise you reading it. It led to the world's first NMR pulse code sequence, and I have used the Carr-Purcell Myboom-Gill pulse code sequence for a nice experiment. He liked the talk that Sir Geoffrey Taylor gave. Sir Geoffrey Taylor couldn't solve the problem, so our friend Purcell thought he would. He began to analyze how microbes could swim. He was very taken with the work of Howard Berg, who was a new young hiree at Harvard, and Berg was looking at how microbes swim. I didn't bring along their video, they don't always play well, but microbes are fast. They dart here and there. Whoops, we made a mistake there. They dart around, they accelerate, but they can't glide. They come to a sudden stop. So here we are. Here's Purcell's applied physics analysis of a little microbe. He gave this lecture on this in 1976. And in 1977, the lecture being so well received, it was published, as is, with all these hand-drawn diagrams. It's a lovely paper. Recommend everybody read it just for fun. There's a microbe about 1 micron size. It's swimming at a few micrometers per second. The Reynolds number is small. That's why it's called life at low Reynolds number. The Reynolds number is the ratio of the inertial forces to the viscous forces. This is like you trying to swim in glycerin. How does this little guy do it? Oh, yeah, you've changed it for me, bless you. Reynolds number is the ratio of the initial forces to the viscous forces. If you are a fish, you swim by inertia. Efficiency is important, and you have a sleek body. You coast. You slide through it. If you're tiny, you can't glide. It doesn't matter if you're sleek and slim or not. It just, you can't glide. There is now a huge literature on low Reynolds flow in water, but nothing refers to the relationship to life. The vast majority of organisms on Earth live in the ocean. Propulsion by the flagellum is by far the most common mode of locomotion on Earth. There's more microbes than there are of us. So if we don't understand that, we don't understand much. But here's his analysis. Things can't swim well. So let's have the next slide. And he tried to work this out. Now, you might think, reading this paper, it was a description of a pond or the ocean. He was too cagey for that. No specific body of water is mentioned. No water molecules are mentioned. It's just an engineering analysis in a theoretical way. There's no temperature, there's no salinity, there's no pressure. It's just imaginary water. A couple of points. In order to get anywhere, it has to go faster or further than you would go by diffusion. See, there's a scale length for diffusion, and he worked it out. That's its goal, to beat diffusion, go from here to there faster than the water molecules move. And he worked out the energy required for this, knowing the mass of the organism, the work it takes, and what he thought was the efficiency of propulsion. And he came with the idea that microbes swim with an efficiency of 1%. That means 99% of the metabolic energy of this little animal is wasted. It's lost as heat. It doesn't go anywhere. This must be the most inefficient organism on the planet. Microbes are known to be very efficient. I think they are. So what went wrong? So Purcell does this, and this paper acquired huge notoriety. So look at the next one. Motion in the oceans, motion on a large scale, is driven by the Navier-Stokes equations. George Gabriel Stokes in 1850, the Frenchman Navier. The Navier-Stokes equations are essential for describing flow in the oceans. All the models you see of the Gulf Stream, of water flowing out of El Niños, all driven by the Navier-Stokes equations. You can't swim that way if you're a microbe. If you throw away the Navier-Stokes terms, you're not left with much. You can't swim by reciprocal motion. If you try and swim in sticky stuff, there's as much pushback when you do this with your arms as there is going backwards. That's his clamshell theorem. So his answer was to work out a propulsion matrix. And he decided the way to do that was to treat the flagellum as a rotating, rigid, helical coil. Next one. And here's a diagram. So this is the flagellum of the bacterium. It is rotating round and round like a corkscrew. There's a propulsion matrix for this. He worked it out, and it's complicated. The analogy there is a little man there in a bathysphere, and he's turning a crank handle like this, and the spiral above him is rotating around. He's trying to make it move, and he's trying to get efficiency. All this is worked out rather well, and people were impressed. The bottom line is, it's 1%. Everybody now believes that. To me, it seems silly. Let's try the next slide. This lecture and this finding drew worldwide fame. It's been put in all the textbooks. It acquired currency among microbiologists working on swimming worldwide. You find all kinds of references to it, comments like, microbes swim. It's not very efficient, but it works well. It's not a complete oxymoron. Move on 20 years to 1997. Oops, skip me back. I inadvertently squeezed this. One more back. I'm going to give up on this thing. Purcell, that great man, that wonderful person, was dying. He's on his deathbed in 1997. What did his mind turn to as he was dying? He went back to this paper. People regard this as a triumph that he was wanting to go back to this problem. The paper was submitted, he died in March 97, the paper was published in the October. His friends and colleagues rallied round and made sure it was published. He did it very carefully, and he came up with an efficiency, a revised efficiency of 1.7%. This was regarded in many ways as a triumph. I have read through this paper several times with a different eye. I think the poor man was troubled. The lecture which he gave, which was a fun lecture to honor the retirement of a friend, had become a phenomenon, more than he ever wanted. And he worried about that, I think. I'm the only one in the world who thinks that. And he thought, have I made a mistake? Have I misled people? Why is it 1%? So he went back and he redid the entire set of arguments. And looking through it, he took pains. It's kind of hard to read. He was painstaking in trying to get it right. The poor man died. He was greatly loved, a wonderful paper, but things weren't going well. Let's go one more slide. The problem doesn't go away. This is a much more recent work. This was published in PNAS in 2006 by a team in Pittsburgh, I think. They got very modern. Optical tweezers. The red cones there are laser beams. The little gray thing in the middle is an engineering drawing of a swimming microbe. And they point out you can do the experiment two ways. The microbe can have its head stuck between the tweezers. Its little legs are trying to swim, but it's not getting anywhere. Or you can pinch it off at the tail, right at the back there, and it's trying to swim its tail. And the flagella are floating around and not getting anywhere. You're just different flow. They did the full-scale propulsion matrix. They analyzed all the dynamics. This is a clever piece of work. These people are smarter than I am. Trouble is, the microbe's not going anywhere. It can't swim properly because the work's not being put to use. And they came up with a swimming efficiency of 1.7%. So after 50 years, it is now if I quote Jane Austen, it is with pride and prejudice a truth universally acknowledged that microbes, in spite of 1 billion years of trying to get it right, haven't learned how to swim properly. Oh dear. That would turn evolution on its head. Let's look at the next slide. Oh my goodness. Microbes are as effective as ostriches in running, swimming, and everything else. The people who have read Purcell's work are stuck. They're flummoxed. They're in a jam. They're in a fix. They can't get past it. The mathematics are right. The propulsion matrix is correct. The laser tweezers are right. If you've not read those papers, you're just going by your biological instincts, then you make a diagram like this. This is a wonderful plot by a French team. Covers 20 orders of magnitude, getting close to Avogadro's number, one of your favorite numbers. It's huge. All the way from elephants and whales down to microbes. It follows a thing called the allometric scaling laws, which relates energy, mass, and size in biological systems. I can recommend this paper to anybody. It's beautifully done, and they relate these numbers to fundamental properties such as the energy from the sun, the size of the water molecule, the strength of the bonds in the myosin fibers, and so on. The little microbes we've got are right down the bottom there. But when you analyze body lengths per second or work or whatever, elephants, racehorses, and microbes come out to be about the same. An ostrich doesn't swim at 1% efficiency. Neither do these guys. What went wrong? Let's have a look at the next slide. Anybody who's awake and knows that we're at a conference talking about water molecules will notice that Purcell had no water molecules in his equation. They were swimming like through air or something. Water was a featureless fluid. It had no molecular structure. The problem is he thought of viscosity as a number you look up in a table rather than a molecular process. He did give one go-away line. He said the reason viscosity changes, it's got one of those activation energy things. And having mentioned that, he thought to hell with it and went on anyway. Let's take a look at the next slide and introduce another hero of mine, Henry Eyring. A wonderful guy, deserves more recognition than he gets. He's old style now. He got the family were Mormons. They were in Mexico as farmers. They were kicked out of Mexico during the Revolution. They moved to Arizona. A devout Mormon all his life. He got his education, where else? At Berkeley. I don't know which lab he was in, possibly with Gilbert Lewis, I don't know. He's done beautiful work. I've used his work to look at things like how the oxygen status of the ocean will change under global warming. Organic reactions proceed faster. He did not get the Nobel Prize. The Royal Swedish Academy gave him the Berzelius Medal, which I thought was a very handsome thing to do. I think everybody appreciated that. Well done. He envisioned the molecular basis for viscous flow, the way water molecules move when they're pushed around by flagellae. It's a nice piece of work. It's not much appreciated today. Let's take the next slide. He thought of viscous flow, the way water molecules move, as an Arrhenius-like reaction. You need an activation energy to overcome the attractive forces that the neighbors have and get to the next step. There's some nice theories about this, and it works rather well. I think Eyring must have been a bit of a pain to deal with. His papers have rather grand titles, the general theory of plasticity, and viscosity, and so on. It's rather grand. So I'm guessing he might be difficult, but the equation's about right. As soon as he'd written that paper, he'd put his student Ewell onto this, nice young man, fix it for water, and Ewell tried to do that. They didn't have very good viscosity data, but they reached a nice idea. In order for flow to occur, you need the individual H2O molecule. The hydrogen-bonded structure of water doesn't work well. And the challenge then is to find the most efficient way to create a population of tiny H2O molecules that would allow flow to occur. If you want to picture this, you're trying to merge onto a freeway in a giant truck. There's cars everywhere. You can't merge into the traffic unless you're smaller. If you make yourself smaller, you can nip into the traffic. It's not a great analogy, but it's something like that. Another hero, let's go to Bell Labs in the 1960s, a wonderful guy called George Walrafen. He died way too early in a car accident in Maryland. I don't have a picture of him. That's Bell Labs as it used to be, a wonderful place. Penzias and Wilson, the cosmic background radiation. Wonderful advances in telecommunications and so on. And he did some careful Raman spectroscopy and came up with this diagram that the tetrahedral pentamer is the most common, most stable form of liquid water. It looks a bit like a methane molecule, doesn't it? You know, there's one in the middle and there's 4 bonds. Rich Sekele knows all about this. This is the most stable, energy minimum, closest compact form of water you can get. A great deal of water in the ocean is in this form. Most people don't know that. There's a part I don't know about, and I'm hoping somebody here at Berkeley can help me. He had a one-liner in the paper. 2 hydrogen bonds must be broken to disrupt this structure. All the other molecules, ones that Rich Sekele works on, you only need to make 1 bond. This seems to be true, but I don't know why. So what do we do about it? Well, I told you I go on a ship. I have lasers. I point them in the ocean. Peaks show up. Now, we are usually measuring gases, methane, animals. Always to one side, there were these nasty water peaks that kept showing up, so we thought we'd better work on them. This is a water band. It's comprised of a whole bunch of molecules. There's an isosbestic point. There's one point in temperature where they cross over nicely, and you need to separate out that big band into several small peaks. We found we weren't doing it very well. If the head of my technical team did it with the conventional commercial software, he got one answer. If our postdoc did it, he got a very, very similar answer, but not quite the same. If our student did it, he got another very, very similar answer, but not quite the same. And if I tried to do it, Peter, step away from the computer. I would, he's my back. What do we do? For all you young people out there, we did what any smart scientist my age would do. We got help from a kid. One of our senior technical staff came to me and said, Peter, I've got a nephew who'd like to work in your lab for the summer. What have you got? I said, well, you have rules against nepotism. What does he do? He's a double major in physics and engineering at Dartmouth, and he'd like to work with your laser stuff. I said, okay, put him in the system. He got the votes. We welcome young Matthew Wojciechowicz, tribute to Mbari's intern program, which Kennedy, who just introduced me, did a marvelous job at spending the summer in the lab. So we deconvolved that mass stuff into 5 peaks. One of those peaks, the little one, can we go back, that's back one slide. The little peak on the left there, you don't bother about, that's an overtone from what we call a libration further on in the spectrum. It just shouldn't be there, it just creeps in. What's left are 4 big peaks due to the water. Okay, the next slide. And 2 of those peaks get bigger as we warm the water, and 2 of them get smaller. The ones that get bigger are due to the single H2O molecule. The ones that get smaller are due to the hydrogen bonded complex. Water is an H2O, H2O5, or H2ON complex. It's an equilibrium, it keeps moving. And if we plot those as an Arrhenius, as a Van 't Hoff plot, we get these beautiful straight lines. Next slide. We need to convert peaks on a graph into molar quantities. Unless you have numbers, you can't do anything. That's never been done before. So again, we go back to the literature. Linus Pauling, another hero of Berkeley, worked out if you melt ice, a certain number of bonds are broken. And from the heat of melting, you can tell how many bonds, and he came up with a number. He thought there was, that the bond enthalpy of fusion was, he knew it was 1.44. That was a pretty good estimate. In 1964, George Walrafen repeated this estimate, and he did an improved version of how much H2O there is once you've melted ice, and the rest of the hydrogen bonded form. He came up with 10%. We stuffed that number into our equations. They have different Raman factors. The polar molecule absorbs light, or gives a Raman signal, much stronger. In fact, 10.8 times more strongly, because it's polarized. We take that number, and we plug it in there, and we can tell the concentration of hydrogen bonded and single forms. The concentration of water in water is 55.5 molar. So if there's 10% as H2O, the concentration of H2O in your glass of iced water is 10%, or 5.5 molar. We know that rather well. This number works out far better than we ever expected. Look at the ocean. Most of the water, by far, is in the hydrogen bonded form. Most people don't know that. Next one. It's not all George Walrafen. A good bunch of it is Richard Sakeley. And here's from his very nice work, Koch and Sakeley, water dimers, water trimers, water tetramers, water pentamers. They're all there. He sees them with very fancy optics. We don't see them with our little laser. Do we know we're there? Yes, I see them all the time. How do I see them? Let's take a look. Next slide. They form methane hydrates. You want to see those pentamers and hexamers? Go down about 600, 700 meters deep out in Monterey Bay or off San Francisco. Take a tank of methane gas down, open the valve, and methane will come into the ocean. It will immediately gather a skin of hydrate. Hydrate is a form in which the methane molecule is encased in a cage of water. That methane molecule is dry. Although it's surrounded by water, it doesn't look like dissolved methane. It looks exactly, it's modified slightly by the cage of water around it. That's what I think locally people call a buckyball. That's actually what we call it's a mix of pentamers and hexamers. 12, obviously 10, mostly pentamers, some hexamers arranged in this nice form, and they stack up to form beautiful crystalline structures. Over there, on the right, that cliff face, it's about as tall as I am. That happens to be off the west coast of Vancouver Island. It's an exposed face of methane hydrate that grew in the sediments. I think a fishing trawler exposed it. It's dissolving slowly as we speak. We fly up to it. Our vehicle is just out of the picture. We shine a laser on that. And I can tell you, just by looking at the light that bounces back, that this is a methane-ethane isopropane structure, mostly methane. I can give you the cage structure. I can give you the dimensions of the cages and so on. It's very nice stuff. There's vast amounts of this in nature. It has been touted as an energy source. It's been called a climate threat. All those things are overrated. It's hard to produce energy, but we find stuff everywhere. But yes, Richard Sakele's pentamers and hexamers are there in abundance. Next slide. George Walrafen had made an estimate of the hydrogen bond strength. His best estimate was 2.6 kilocalories per mole. We felt in order to make progress, we needed to try and make our own measurements. So we did it classically simply. We went out to sea. There's the apparatus. There's a green laser passing through a little cell. There's a heater in there. We put the laser through it. We measure the water. And we know the ratio of H2O to H2ON, the other forms. We warm the water. Free water goes up. The bonded water goes down. And we get a line. We calculate the molar concentrations at each step along the way, and we carry out a very simple Van 't Hoff plot. And from this, we got an estimate for the hydrogen bond strength of 2.624 kilocalories per mole. I think we did pretty well. George Walrafen was right. And this is very much homemade science, a heater, a laser, some tables, and some clever thinking. It works well. Next slide. Okay. Getting back to viscosity and the way that microbes have to swim and whether Edward Purcell was right. He didn't have good viscosity data to work on, although this had been published before. US Navy scientists measured it. This was an apparatus they built, looks like a giant gun. It went inside a giant pressure tank. Electrically, they released a ball bearing. It rolled downhill, and from this you can get the viscosity. It was quite the gadget. The US Navy paid good men, good money to watch a ball bearing roll downhill. What did they find? Here is their paper. If you look at the next slide. The experiment was beautiful. This paper is a goddamn disaster. I looked at this and threw it away. I looked at it again and threw it away. And what the hell is going on here? They got mixed up. I couldn't quite tell what was going on. It took me a long time. I finally realized they measured temperature, measured 7 temperatures. They managed to get 15 points on the graph. How did they do that? Well, they thought there weren't enough to give them a good fit, so they did some interpolation and added some just for good luck. Students out there, if you ever feel a graph is looking a bit thin and you're tempted to add some more data points to make it look better, don't. Okay? It's a bad idea. And they came up with this crazy idea that the activation energy was temperature dependent. They got an approximate number. This graph is a disaster. So what the hell do we do with it? So we had to reshape all their data. The next slide. Am I running out of time? Good, okay. We just did the old-fashioned way. We went to our friend Henry Eyring. We used his equation. We took this data, which we had to rewrite, and we made a good old-fashioned plot, just like Henry Eyring would have told us to. Look at this slide next. And we got this beautiful straight line. The correlation is 0.99999, okay? At any one pressure. Then you increase the pressure, measure it again. From the slope of that line, you get the activation energy. It got less. Higher pressure, it got less. Higher pressure, it got less. This is going backwards. Viscosity isn't, the activation energy is not supposed to go down as you increase the pressure. What the hell is going on? Next slide. We couldn't figure this out. So we went to sea and did our usual thing. We measured the strength of the hydrogen bond in water at different pressures. The upper line, that's in Monterey Bay, fairly shallow work. We had the heated cell, the laser was sitting inside the cell. We got good results, and that's the line. That's the change in the activation energy viscous flow, a change in the strength of the hydrogen bond, with pressure. Down below, it's a different line. That's because we had a different experimental configuration. The laser was outside the cell, so a bit of native seawater got in. We'd been down south, investigating an alleged chemical munitions dump site. What a mess. The seafloor's chaos down there. We had a spare day, and I said, let's go and have some fun. We'll do a really deep ocean experiment and measure the strength of the hydrogen bond at depth. And we got this second line. Those two lines should meet up if we'd done it right. So the activation energy of viscous flow and the strength of the hydrogen bond both go down with pressure. Let's look at the next slide. What's the effect of pressure on seawater? This comes from a paper by Campolat et al. I don't like the paper. It's awful. But I love the diagram. When you apply pressure to seawater, you push molecules closer together. What's the easiest way to do that? These giant tetramers, think of it like a giant camera tripod. You try and get that in a closet or in a car trunk, they won't fit unless you put the legs together. You nest the legs, just like that. When you apply pressure to seawater, you force George Walrafen's pentamer legs to nip in together. Nobody appreciates that much, but it's very powerful. Keep going. Why does the activation energy of viscous flow decrease with pressure? Because the hydrogen bonds get weaker with pressure. Why does the hydrogen bond get weaker with pressure? Nobody knows. Let's guess. Let's try some high school chemistry, Le Chatelier's principle. The chemical system reacts to oppose change. You compress water, you increase pressure, it doesn't want to compress anymore, hey, get back. If it kept on getting more and more compressible, you'd have a blob of super dense water at the bottom of the ocean. So it resists compression. What that means is there's less George Walrafen at the seafloor and more Richard Sakeley, because those forms he builds don't want to squeeze together. That's what's going on. Can this be true? Next slide. We see it all the time in the speed of sound. Sound speed is a mess. It was hugely important in the Cold War when we were tracking Soviet submarines. Ocean acoustics was a big deal. The problem with sound speed is they make a mess of it. You ask an acoustician, what controls sound speed? It's temperature and pressure. Sound doesn't propagate through temperature. Sound propagates through water molecule, the molecular structure of water. So all we did was to take sound speed and plot it as a Van Hoff plot. 90% of the variance is explained this way. It's controlled by the equilibrium between the Van Hoff, between the pentamers and the single molecules, and we can fix this. The equation, sound speed equations got up to 125 coefficients. We got it back down to 25 with a piece of elementary chemistry. Next slide. Does that work? Yes. The sound speed minimum in the ocean is caused because as we go down, it gets colder. Colder, you get more pentamers. And however, under more pressure, they disappear. The sound speed minimum is how whales communicate over thousands of miles. It's important for tracking submarines. And it's related to the strength of the hydrogen bond. And you can map that rather well. Next slide. Can we solve Purcell's propulsion matrix? No, I don't know. There's a science of molecular dynamics. There's a science of fluid dynamics. There's no science of molecular fluid dynamics. What happens to the water molecule? The microbe swimming, water rich in the single H2O species is coming off it. It meets the rotating flagella and it gets some angular momentum. What happens then? We don't know. It's almost like, and this is fantasy, it paints a stripe on the flagellum. One more. The advantage of breaking hydrogen bonds to swim is that you shake off your environment. Purcell was worried about that. In a viscous solution, when you stir, you carry everything along with you. If you break the bonds, you get rid of it. That's why they do it. Next slide. How does a microbe swim then? We can't give you the exact answer, but it's like this. Imagine you're a student and you walk into a giant crowded ballroom and you want to get through it. If you're a giant fish, you don't care. You smash through it, bodies going everywhere. If you're a little microbe, you can't do that. You're too small, you're too fragile. So you walk along and you say, excuse me, step aside, excuse me. You break one hydrogen bond at a time. You part the waters gently. That way you don't smash yourself up. The strength of the bonds, which control your motors, are not that different to the strength holding the crowd together. That's how they swim. Finally, one more. An article from that famous scientific magazine, the Wall Street Journal, by Frank Wilczek, appeared at the end of the year. He was trying to explain the discovery of the Higgs boson. Very complicated, too tough for most readers. So he led off with a parable. Let's imagine an ocean, a planet, which was all water, no land. And the only living things were fish. So they developed some intelligent fish. The fish finally managed to discover Newton's laws. Things move, they're pushed, there's forces, tide, and so on. But a few intelligent fish get together and say, what's this water stuff? So they boil some water, they do some spectroscopy, they discover chemical bonds, and they find water molecules. Hooray! I love this. So I wrote to Frank Wilczek and said, if you've been reading our papers, we didn't boil water, we just warmed it, but we did do spectroscopy and so on. I got a charming reply. It was funny. He said, use this article anywhere you want. But I told him the Wall Street Journal needs to hire better cartoonists. You can't light a Bunsen burner under the sea, and water doesn't boil until it gets about 300 degrees and you exceed it. So I said, Frank, I don't think you proofread this very well. You need a better cartoon. Okay, that's it for me. Thank you all. Thank you, everybody. You're a lovely audience. I really appreciate the chance. Thank you, Bengt. Thank you, everybody in Göteborg. Thank you, Laurie. Thank you, Omar. Everybody here. Thank you, Kennedy, for the introduction. My colleagues. Thanks to David Packard, a wonderful man to work with, for building the lab. And thank you to my wife, Hillary, for putting up with all this. So thank you all.